Okay, so we're going to go ahead and keep talking about the chromoalveolates and the straminopiles. We left off last time with the diatoms, so we want to keep talking about this group and look at what's unique and interesting, the diversity of them. Why are diatoms important is going to be a big question for us to keep in our heads as we go over the topic here, or this group. All right, so first thing to mention, diatoms are single-celled. The brown algae are multicellular, but with the diatoms, they're all single-celled organisms. They are all going to be photosynthetic, which means they play a huge role in the producer base of, let me add this there, marine ecosystems. Okay, so very, very important when we talk about marine ecosystems. Big part of the producer base. So here's a big hint. Let me put the little star next to this. What I would encourage you guys to do is make notes. Which protists play a role in the producer base of the marine ecosystem? So yeah, you're going to need to jump back one of those lectures or look in your notes and go, what other protists did we talk about that also is a producer within marine ecosystems? Huge issue. Huge issue. These will come back to us. So red star here, go back a couple pages in notes, start listing out producer base of the marine ecosystem. Keep aside, maybe a sticky note in your sticky tab in your notes or something. Track these guys. All right, so to be a producer, they're going to be using chlorophyll A and chlorophyll B. Oh, I'm sorry, chlorophyll A and chlorophyll C. Make sure if I can spell chlorophyll right. Chlorophyll A, chlorophyll C. And then they're also using the carotenoids as their photosynthetic pigments. So we have this color diversity, browns, tans, some greens, some yellows, those colors within the diatoms. Looking at the structure, they have this double shell of silica. So silica is a hard structural material. So imagine a petri dish. Some of them have that shape. Imagine a petri dish, hard, durable, stable, structural integrity. And then within the petri dish, they have hundreds and hundreds of these little pores or slits known as raphe. So, and I'll show you that in a minute here with the next picture. It's much better. But that's the general shape. Now, they can be round like a petri dish. They can be tubular. They can be whatever you want to call this, teardrop shape. I don't know what that is even called. But they all contain that double shell of silica. Now, in order to move around, they will glide on a slimy secretion that they produce, or they can just be pushed around by water currents. It's not like they can resist the wave of the ocean pushing them around, moving them around. One of the big areas we'll see these guys in our daily lives is this stuff called diatomaceous earth. And I'll get into that a bit more in a minute here. All right, so big general features of the diatoms. All single-celled, photosynthetic, producer base of the marine ecosystem. Very important group when we talk about marine ecology. If diatoms crash, your food chain crashes. It's a big issue. Reproduction is primarily through asexual reproduction. They just split themselves in half. Occasionally, they will reach a point where they have to go sexual reproduction. So this is how this is going to work. Let me make this guy a little smaller. So here's your original diatom. It's tooling around, doing its thing, and it says, oh, time to reproduce. So what it's going to do is it's going to pop itself in half. That's supposed to be the top of the shell, and then this is the bottom of the shell. Now, one side, top or bottom, is going to be the larger one because it's going to sleeve together like a petri dish. So what happens when it reproduces is the new part, because it splits itself in half, the new part slides inside or develops inside of it. Actually, let me make that. I'll make that in red so it's easier to understand, hopefully. So this new half is actually a little bit smaller than the original size. Now, does its thing, and then it decides, oh, we need to reproduce asexually again. So the new piece that's going to go inside the red part of the shell is again going to be smaller. So what we will see over time is that the size of the diatom will get smaller and smaller and smaller as it undergoes asexual reproduction. So it's just this reducing its size in order to reproduce asexually. At a certain point, it goes, oh no, no, we're getting too small. And what it will do in a following generation is it will take and produce gametes that will fuse together to create a single cell, diploid, full genetic material that will grow back to the original size of the original diatom that you started with back up here. And then the process repeats itself. So there's constantly diatom reproduction happening across the Earth's ocean. So it's crazy. Their diatoms are literally everywhere in the marine ecosystem. As I mentioned before, they are a major component. Oop. Oh man. There we go. Major component of marine phytoplankton. The walls of the diatoms, or the diatom shell, equals this stuff called the diatomaceous earth. So when the organism dies, the shell remains. That hard, rigid, structural shell remains because it's made of silica. That's the same kind of stuff that's in glass. It's hard, durable. It remains. We also can use diatoms as index fossils, meaning they tell us a history or age of the Earth's history. So if you see accumulations of diatoms, you can say, oh, this part of the ocean is this many hundreds of millions of years old. These don't accumulate quickly. It takes time for diatoms to build up in any substantial numbers. But one application for us, well, there's multiple, and I want you guys to look into what are uses of diatoms in our daily life. But one application is filtering agents. So imagine this tiny little thing with all these tiny little pores, pool filters. Sometimes we'll use diatomaceous earth. So water goes through that and it filters out tiny little particles, bacteria, debris, stuff that you don't want floating around in your pool. So it's a great filtering agent. So again, lots of different uses for these. I want you guys to explore some uses beyond pool filters. Where else in our daily lives do we come across diatomaceous earth and the diatoms? So anybody working towards veterinary medicine, you will probably be exposed or have experience working with these or use these for different things in the veterinarian field. So, okay, all right, so that was our second group under the Straminopila or the Straminopiles. Third group are these things called the oomycetes. Now, these guys are just a weird, goofy, unappreciated group. So historically, these have been called water molds. They are not a fungus. I'm going to put that in here. Not a fungus, even though they're called a mold. That is the traditional general name, which does not accurately describe them. Unfortunately, they're often parasites, or these, we classify them as sapropods. They break down and decay dead stuff, or they parasitize living things. So the picture you're looking at, all those little hairy things, that's the water mold. The brown tannish stuff, this is an exoskeleton of the insect that they parasitized and literally have killed. So when we look at them, they have a mobile stage in their life cycle called zoospores. And those zoospores have two unequal flagella on them. So flagella with hairs because they're Straminopila, two different length ones. Those are your water molds. Reproduction is sexual or asexual, where they can fuse gametes or they can just split themselves into two, and now you have more water molds. Diversity of habitats and environments. They can be found in aquatic systems. They can be found in terrestrial ecosystems. They tend to need moist, wet environments. Not real good in the desert, but if you have enough moisture, then you can see those guys in those environments. So where we may have experience or exposure to these guys is fish tanks. So if you guys have had a fish die and all of a sudden it gets fuzzy and you're like, oh, that's a fungus. Probably not. It's probably a water mold, an oomycete. So be careful. So what we see, those little fingers, those little fuzzy things, are called hyphae. And that's what enables them to increase their absorption of nutrients. Are the little hyphae, or these filaments that they contain. Huge ecological impact. So again, fish tanks, if you guys have this and this shows up in your fish tank, you're going to have some problems, because even if you take the dead fish out, get rid of it, those may still be, the zoospores may still be swimming around your fish tank, and they can parasitize your fish in the future. So you need to do a heavy water change, try to get all that stuff out as possible and keep an eye on it. If the fish are healthy, they shouldn't have problems. But if your fish gets stressed, these guys can be opportunistic, and all of a sudden, boom, now they're starting to parasitize your fish and can possibly kill the fish. We see them also responsible for amphibian decline. They will parasitize the eggs of many species of amphibians and lead to some problems with amphibian populations. So unfortunately, another thing that's beaten up on our poor amphibians out there. And lastly, historically, these things played a huge role. Anybody who is familiar with the Irish potato famine in Ireland, there were two, 1845 and 1847, which drove a large number of Irish immigrants to the United States. It's due to oomycetes or water moles. They had lots of rain, excessive rainfall in Ireland those years, and the potato harvests were decimated because of this water mold. And the problem is, if it's in your fields, you can't get rid of it. The next time you have a wet spring, oomycetes like that. They like moist, wet environments. They're going to pop back up, parasitize the foods that they can find, and lead to more problems. So anybody with Irish heritage, your ancestors were immigrants who came to this country on a boat, legally or illegally, whatever the case is. If you have Irish heritage who came to the United States in the 1840s, it was most likely because of these things. People were starving to death in Ireland. They came here looking for a better life and came to the United States to start a new life. So lots of connections within history, ecology, etc. to groups like this. So big picture, when they reproduce, they can do sexual and asexual. The asexual is pretty simple. They just produce little zoospores. Those things can sit in this cyst stage, which is a dormant stage, and just hang out and wait. And then they germinate, and now they're back and sending out little filaments of hyphae everywhere to decompose stuff or parasitize things. If they're going to go asexual, they produce the different zygotes, or I'm sorry, different reproductive structures, eggs and then the oospores. Those guys will fertilize, create the haploid, diploid structure here, the 2N structure, and then that's going to produce these little zygotes that will then germinate, develop the hyphae. The key though is when you're producing sperm and egg and they fuse, you're changing genetic combinations, creating new genetic diversity. So, okay, all right, so the last thing to wrap up here with will be the review slide. Okay, so let's go pop down to that review slide, see what you guys have in your heads, see what you can do with the review here. All right, so without using your notes, let's see if we can do this. I'll put the green color here. All right, what is the key feature of chromoalveolata? Can you list out that key feature right there? What feature is required to go to alveolata? Do you know that trait? What feature goes there? What feature do we need to go to straminopila or to straminopiles? What feature goes here? And then once you're in straminopila, what makes brown algae unique? What makes it different than the others? What makes the diatoms different? And what makes the oomycetes different? What are their key traits or features that you could use to say, this makes these guys different? Okay, so if you can do this without your notes, awesome, you're hitting a home run. You guys are going to do phenomenal here. If you are not sure, go back and review and review and review and just keep reviewing, work with these review slides to make sure you guys get this under your belt. Okay, all right, so we'll pause it here and then move on to part 3 where we're going to talk about the archaeoplastids.